So, this lecture I will cover few databases, mainly the DNA databases and the protein databases, right. So, in the last class we discussed mainly about the DNA. So, I will move on to little bit about the DNA databases, right. So, it is very important to get the sequences of the nucleic acids, right. So, at the same time there are different types of databases have been developed. For example, so one is the DDBJ, this is the DNA data bank of Japan, right. So, this is in Japan, and the second is EMBL. EMBL is a European Molecular Biology Laboratory. So, they also developed this collecting nucleotide sequence data, and GenBank in US they also started to collect. So, similar time, because several people they think in a similar way, right. We cannot say this is very unique that only we will do, because science is open and research is open. So, it is very competitive, right. If you have some information, immediately several people will think in the same way. This is the reason we need to be very fast and very accurate, and we have to provide reliable data. So, they started collecting the data, and then they also developed some tools to analyze the data, right. Then what happened in this case, each databases they have different types of data, right. So, in this case there is some discrepancies. So, users have to access all the databases, right, because sometimes you get the data from DDBJ may not be available in EMBL, sometimes you get the data from EMBL may not be available in GenBank. So, for getting a data set they have to check all the databases. Second aspect is the search options and the display options and the format may not be same. If it is different, then again we have to rework, right. So, then they made a consortium, right. So, they joined together using this science information network. So, here is the DDBJ, right. So, NCBI and we have the EMBL. So, joined together. So, they share the data. So, now the data what you get from DDBJ you can from EMBL or from the GenBank. So, wherever you use or access the database you will get all the information, right, fine. So, now I will just demonstrate a bit about these databases. Okay, DDBJ, this is in Japan, in Mishima, Japan. Okay, this is a beautiful city in Japan. You can have this Fuji mountain and all, fine. So, here they have different options. You can submit the data, and you can search and analyze the data, and they have the facilities, and also you can FTP the information, right. So, this is the website ddbj.nig, the National Institute of Genetics, this is the institute that maintaining the database, and ac.jp. So, you can access this website and you can get the data of the DNA sequences, fine. So, this is a growth, okay, this is they started long time ago. So, it is in 1999. So, this is the in January, this is release number 36. Before that they started to initiate the database, and this is a current status. So, this is 2016 106 release. So, this we have the 2.5 trillion nucleotides as of June 2017, this latest update, and about 875 million sequences. So, you have plenty of sequences for the analysis, and we look into the organisms as we expected. So, we have the topmost human and followed with the mus musculus, and then you can see E. coli and other organisms. So, you can this is the topmost organisms in the DDBJ, fine. This is nucleotides, this is the number of entries available for each organism, right. So, now how to get the data? So, there are various ways to get the data from DDBJ. The first condition is the very simple search, that is a quick search. If you want to get the data for any of these your DNA or RNA, so here I put the Homo sapiens mRNA for this protein, right. So, if you search, right, then you will get the link, right. When you click on this access number, right, that 36833, right, then you get the complete data. So, now see what are the contents, right, which are available in DDBJ. So, first you give the name, right. So, I give the data. So, here is the Homo sapiens mRNA 315 base pairs, right, and the source is Homo sapiens, and here they give an access number specifically for this DDBJ. So, this access number, and they give them keywords, right. So, in this case you can search the data based on any specific keywords, right, and the others who are the one who sequence this data, and the reference, you give the complete reference, and they give the PubMed index, they give the link to the PubMed. PubMed is literature database, okay, that I will explain a little bit later, right, and they give the data of number of A's, T, C and G. So, here the number of A, number of C, number of G, number of T, and this is a complete sequence, right, 315 10 15 base pairs, right. So, in this sequence, right, so what is the AT content? How to get the AT content? AT plus 76 divided by 315, right. You can easily calculate this is number of A's, this is number of T's, and you can calculate the AT content, right. So, they give the translated protein sequence also, right, the fine. So, the advanced search, so they give a lot of options, right, some of them are familiar, some of them are not familiar. So, in this case they give the ontology of each terms, right. If you click on this one, you will get the details of, right, the specific term. So, here I put the sequence length 400 to 500, right, and organism human, right. You can use any of these search options to get your sequence, right. Now, if you give these are the list of sequences, there are so many, for example, there are 26,506 entries between 400 and 500 from human, right. So, then if you click any of this axis number, you will get the full data, right, I discussed earlier. So, they give the definition, they give the keywords and the journal name, right. So, this is the translator protein sequence, right, this is the NIY. Last class we discussed about the coding, right, is the one codes for a protein amino acid, right. So, this is a protein sequence, this is a DNA sequence, you will get the protein sequence, fine. Now, the DDBJ they have the option to upload your data. If you have niche sequence, right, you can also upload the data, right. So, either you give the single sequence or the multiple sequences, right. So, they receive the data and they first validate, and if all aspects, if the data is clear, then they will include your data in the database, right, that is fine. Similar way, so now we discuss about DDBJ, similar data are maintained in the GenBank and EMBL, right. So, what is GenBank? How many of you use GenBank? Fine, right. So, what is GenBank? Right, it is NIH genetic sequence database, right. They provide the collection of the information regarding the DNA sequences, right. So, they give all the information, this is the website. So, you can see the GenBank, you will get the information regarding the sequence available in GenBank, right. It is developed by NIH, right. So, NCBI they are maintaining this database, right, and all the details they published in your database issues NAR, because NAR nucleic acid research is the journal as I discussed earlier. So, they publish the details about the databases and you can get all the information regarding that, fine. So, this is the contents of GenBank. So, like we discussed in the DDBJ, so GenBank also a similar type of content, right. Maybe we can look into this website and get more details, just I will go through quickly. So, it has the definition, because this is yeast, so this is Saccharomyces cerevisiae, right. So, this is access number, right. Here you give the organism and with all these names, right, the reference and the features. So, here they give the various divisions, they give the primate sequences and rodent sequences, mammalian sequences, they have the various conditions, the various classifications they given this separately. So, if you are interested in any you have the specific sequences, for example, viral sequences, right, they give the database data for the viral sequences, they give various classifications, right. So, then again they give the data about the literature and what are different features and the coding sequence. So, what is the coding sequence? Translate proteins, right, the nucleotides that correspond to the sequence of amino acids in the protein, right, how they translate. So, this give from which to which. So, they put n .m means from n to m, for example, 687 to 3158, okay, so this is the coding region. Then if it is a partial one, they put the less than symbol, right, here partial at the 5 dash end, then if it is greater than symbol, they put partial at the 3 prime end, right. So, they give the information, right, they also give the complementary strand information, right, and the translation. So, here this is the search option available. So, you put the keyword here, right, if you click on this go, right, and then you will get the list. So, they give you different types of information, not only the nucleic acid sequences, they give the data about the how many literature in the PubMed, right, how nucleic acid sequences or the GST sequences, right, protein sequence and so on. So, if you click on the nucleotide, there are 19, right. So, now if you get all the 19, these are the 19 sequences you get from the GenBank. Then if you click on this first one, right, then it will show the details, right, fine. So, now you get the data from the GenBank. We discussed about the DDBJ, I have discussed about the GenBank. So, another one is EMBL. EMBL is maintained from European Molecular Biology Laboratory, right. So, one is in US, one is in Europe and another one is in Japan, right, because they have the very good fast computing facility, internet facility long time ago. This is why first they developed there, right. You see most of the databases called PDB, Uniprot and all these databases, right, they have the consortium between all these developed countries, because internet came out there first and they had very fast computing facilities and all. So, they started there because people are trying to use that, right, because India we got very late, right, because when I visited Japan in 1997, they asked me to do with all the everything with email and all, but we have only fax at that time, right, but currently it is synchronized, right, whatever we get in the developed countries we immediately we get in India also, right. This is the reason why almost most of the early developed databases, right, everything is from US or in the Europe or in Japan, fine. So, EMBL, so they also start with the database for the nucleic acid sequence database, right. Here the main sources are the DNA and RNA sequences, right. So, they also accept direct submissions, they are also curating the data, right. So, mainly from the genome projects as well as the parent applications, right. This is the website for the EMBL, right, you go to eba.ac.uk and you go to EMBL, right. So, this is the website, right. So, now if you go to the EMBL, this is the statistics, right, similar to DDBJ now correctly, see the mainly this is the organisms, right, Homo sapiens is the most, right, and the followed by the other organisms. Here also this is by entry count and with nucleotide count. So, you can see the similar level of the statistics, fine. Okay, now go to the database entry. Here also if you see the similar to the GenBank or similar to DBJ, you see the names and the PubMed entries, right, and we have the coding regions, this is the protein sequence and you have the DNA sequence, right. This has 1859 base pairs and you can see the contents of A, C, G and T, right, fine. So, EMBL also have search options, right, you can search the same Homo sapiens GAPD RNA, right, glycerol nucleotide, right. So, now you go there, so they have various options, right. If you give all the results, these are the various results for the EMBL. So, now you have various format to check whether the EMBL format, if you click on EMBL format, right. So, it gives the EMBL format, okay, this is the contents of this particular database, right. We have the protein sequence and the DNA sequence, fine. So, if you use the DDBJ, right, or use the GenBank or use the EMBL, now you can get the data for any of the all the nucleotide sequences, then you can use the sequences for further analysis. So, I suggest you to look into databases and use the options available in these databases and then try to get the sequences from different organisms and see whether anything is different or the same, and also you can calculate different properties, right. Last class we discussed about various properties from the nucleotides, right. What are various properties we discussed in the last class? Flexibility, stacking energy, right. So, depending upon the dinucleotide or trinucleotides, right. So, we classified the nucleotide sequence into overlapping segments, either dinucleotides or trinucleotides depending upon the availability of data, then you can calculate the average values, right. This will tell you how far this particular sequence is stable compared with the other sequence or not, right. So, there is a database called dinucleotide property database, right. Last class we discussed only few properties, right, mainly the flexibility or rigidity or the base stacking energy or hydrogen bond, right. So, this database it has more than 140 features, right. This is available in this website DiProDB, right. So, these are the various information available in this database. Okay, here they have a twist, stacking energy, rise, bend and so on, right. Here these are the various dinucleotides, you have the property names. So, we can analyze various features. That means, because we do not know which feature is important for the flexibility, which feature is important for the binding affinity, right. So, you can use any of the features and try to understand why this is important and what properties are important for the binding affinity, or if any mutation is causing any diseases, you have the mutation data here, right, then you can relate with this disease information so on, fine. So, this database is available in this website, right. So, these are downloadable. So, you can download the data, right, for all the features, and you can use these features to understand the different DNA sequences, right, fine. So, till now we discussed about the database, different types of databases, right, the collection of databases, right, and mainly for the DNA. What are the databases we developed for the DNA? DDBJ, EMBL, GenBank, and this nuclear property database, right. So, now we will discuss about literature database, right. What is literature database? And discuss data about the published articles, right. Earlier days, right, few decades ago, they started to have some abstracts like chemical abstracts, physical abstracts, right. So, in this case, this is a very big volume, very small letters, very difficult to read, that even I am not sure they published, maybe it is in the computer readable form, right. They publish in the chemical abstract and physical abstracts, they are the very famous abstracts. Then he started the science citation index, right, they started to rank the different journals, answer different papers, and see how many citations each article or each journal, published articles in the journals are cited by other authors. Then PubMed is a widely used database for the life sciences, right, they include mainly life science papers, not the physics or chemistry, right, from several leading journals, right, with the reference to the all the papers published in the literature, right. Okay, so I will explain about PubMed because it is widely used database and we get almost all the information relevant to the biology and as well as for the medicine. So, if you want to get the information regarding DDBJ nucleic acid sequencing database, so you want to get the article published about this database, if you search the keyword, right, so it will show you the all the data, right. From this you can get the, this is a collection because this also includes the DDBJ, this is listed here, and you can see so DDBJ progress report and various other information, right. They listed other articles also because they also link with this DDBJ, this is the reason why they get the other articles, right. So, we got the DDBJ progress report, right. So, then you can get the data. So, when you display the data, there are various options, whether you need the format, the summary or the abstract or any other formats, and how many items per page, 5 or 10 or 20, and whether you can sort, whether you need the first author or the journal name or the recently added and so on, right. So, when you apply this, right, then you can get the information, right, this is what we need, right. So, they ask for DDBJ nucleic acid sequence database, right. So, here we have DDBJ, right, so you will get the data. But if you want to get the full text, you click on the full text, then you will get the complete paper because this is the title. If you click here, you will get the abstract, and if you click here, you will get the full text, right. Then you have the related citations. What is the meaning of related citations? The papers which are similar to DDBJ. So, you can get the GenBank, you can get the EMBL, also other databases, right. So, you can get the different entries, published articles which are related to this DDBJ, right. Okay, so, this is the website for the PubMed and you can access this website to get the literature database, right. So, now you get the click on the DDBJ progress report, you get the abstract, and here you can see get this full paper, either you get this Oxford University Press, this is the published NAR site, or you can get the PubMed Central, right. What is open access? So, they put is open access, right. What is the meaning of open access? You can use it freely, right, because to publish an article, publish a journal issue, so it involves lot of cost. Who will bear the cost? Two options. One option is the subscribers, right, they have to bear the cost. In this case, others do not have to pay anything, right. We have to publish the article when it is accepted with the review and other procedures. Publishers to publish. How to earn the money? Because we have to subscribe the journals. When you subscribe, you have to pay. Those who want to read these articles, they have to pay, right. This is how they maintained till a few years ago. In open access means the others will pay, right. In this case, the readers do not have to pay, because others will pay the charges and they tell the publishers to make it open. So, anybody can access this article. So, this is called the open access, right. There are two options. Some journals they can only open access, some journals they can only by subscription, some journals they have the option given to the others. Others can choose whether you want to make it open or not. If it is open, you have to pay, but it is available to everyone. If it is not open, it is not available to everyone. So, only restricted users can read the article, right. So, depends on the authors, they can make it as open access or not. Fine, this open access means you can use it, right. So, you can go with the full text, right. Now, this is a PDF format, right. So, what is PDF format? Portable document format, right. In this case, you can get the full paper, you can read in that format, then you can read it like print, right. Here you can read it online, right, but if you give the PDF format, this just kind of print, like you can reading in a journal article, right, you can do that. So, now what is related articles? If you look into this click on related articles, so it show all the articles which are relevant to your search. So, for example, this is your search, right, the main article. If you go to related citations, these are the other papers which published in the literature which are related to the article which are our query article, right. You get all the related articles, you can get that. Fine. So, what are the various features of the PubMed, right. So, you can search with any keywords, you can get data from the latest article, right, and search the letter entries to give the articles which are relevant to the later articles, and also you can use some stamps to get any of these articles. You can use with the get with the page numbers, you can with the journal name, you can search with authors and so on. So, main difference between current level PubMed and previous years was, previous days unless we get the article, unless we read the article, we cannot cite, but currently if you type any keyword, you get all the articles. So, main problem is currently if you write a manuscript, just you type the keyword, get all the papers, they do not read the full paper, some people they do not read the abstract also, right, they do not know the others, just they take for topmost 5-15 and cite everything. Earlier days the original authors got proper credit, because they do not read all the papers, only the important papers we have to send a request and they will send the reprint, then we read, then we understand, then only we cite. Number of citations if you see is very less, because all the papers almost read by the others, but currently they cite hundreds, but they do not read any of the papers. That is the difference between the advantage of this PubMed, right. Very easily the others get the information. Fine. So, this is another database called disease database, right. This is the 3D Insight is a database which contains various information regarding the protein sequence and the thermodynamics and visualization and so on. They also added some of the information regarding the diseases. So, how about the mutations? They change the mutations, right, they cause any of these diseases. They obtain the data from the other databases and they develop the database for the diseases. This is another database for the protein function database we developed few years ago. So, here it will tell you important residues which are performing different functions in membrane proteins. So, we have the search options and we have the display options and we get the results. For any mutations you can say whether drug resistance or what is the function of the protein, as well as how any specific mutation which alters the specific function or not. Likewise, if you look in literature, the very databases based on the protein sequence, protein structure and the thermodynamics, diseases and the literature and so on. So, you look into this nucleic acid research website, you will get the information regarding all the information, so that you can use it. Thanks for your attention.